Hello guys and welcome back to Excel Academy. So in the previous lecture we talked about nucleosome sliding, injection and replacement, and also we talked about nucleosome positioning. So now we're going to see how exactly these histone tail gets modified, what kind of modification is taking place. So earlier we have discussed that there are certain modifications like acetylation, methylation, phosphorylation. So all these groups are added, and these groups when they get added to the histone tails, they actually change the function of the nucleosome. So let's see what are these modifications. Amino terminal tails of the histones are frequently modified. For example, let's talk about these. So lysines in the tail are frequently modified with the single acetyl or methyl groups, whereas in the case of arginines, it is found to be modified with 1, 2 or 3 methyl groups. So we can have in the case of lysine 1, whereas in the case of arginine, we can have 1 to 3 groups of methyl groups which can be modified with. Then serines and threonines and sometimes tyrosine also, because they have got the OH group in the functional group and they get modified with the phosphate group. So in certain cases, ubiquitin and sumo molecules are also being added, and then they are actually targeted for proteolysis. Okay, so we know that whenever ubiquitin molecules are added, the proteins are meant for the proteolysis or the degradation in simple words. So guys, you need to remember all these things, like which are the amino acids which gets modified in the histone tails. So till now we have seen it is lysine, arginine, serine, threonine and tyrosine as well. Okay, so these are the groups which they have shown. So here you can see that all the histone proteins are having the histone fold domain over here. And now let's see the modification which it actually takes place. So in the case of H2A, you can see that phosphorylation and 2 molecules on the lysine group, right? The acetylation group is possible. Then in the case of H2B, it is mainly the acetyl group and the 1 phosphorylation. Again, on the lysine, you can see the acetyl group has been added. Then coming to the H3, so you can see that compared to the all the histone proteins, H3 is having the maximum modification, right? So it has got both methyl group, acetyl group, phosphoryl group, everything is there. And then comes the your H4 group. So H4 protein. So again here also methyl group, phosphate group, and acetyl groups are getting modifications are being done with these groups. Now one thing to notice over here is that on the H2A and H2B, you can always see another group, that is duplicated molecule. Okay, so these molecules, they actually target for the protein degradation whenever these molecules are being added to the any protein. So what are the important sites? So these specific modifications, they are always associated with different cellular events. For example, acetylation of lysine at positions 8 and 16 of H4 is associated with the start of the gene expression, whereas acetylation at lysine 5 and 12, it marks the newly synthesized H4 molecules. इसका मतलब क्या हुआ? अगर आपका 8 एंड 16 पोजीशन में एसिटाइल ग्रुप हो रहा है तो इसका मतलब वहां पे कोई जीन एक्सप्रेशन स्टार्ट होने वाला है. अगर 5th एंड 12th पोजीशन में हो रहा है लाइसिन के, इसका मतलब दैट H4 मॉलिक्यूल इज न्यू. ठीक है, वो नया सिंथेसाइज हुआ है. Now, if suppose methylation is happening, then in the case of H3, it is associated with the gene expression. And then if suppose it is the methylation of lysine at 9th and 27th of H3, then it is associated with the transcriptional repression. यानी कि वहां पे ट्रांसक्रिप्शन नहीं होगा. ठीक है. So these are the enzymes involved: histone acetyl transferase and histone deacetylase. So from lysine, you can see that acetyl group has been added. And then coming to the phosphorylation. So phosphorylation, we know kinase and phosphatase are involved. Okay, so you can see that on serine and threonine group, you have got the OH in the functional group. So this H is being replaced by the phosphate group. Then histone methyl transferase and histone demethylase. So lysine group, it is getting modified, and see, you can see that either you can have 1 molecule or 2 molecules or 3 molecules of methyl group present on the same lysine molecule getting modified. Then on the arginine group, again same enzyme, histone methyl transferase, you can see over here also. So NH2, it becomes 1 methyl group, 2 methyl group is there. Or you can see the asymmetric. So again here also you can see. So either you can have the symmetric methyl group present, or you can have the asymmetric methyl group present. That is, एक इस तरफ है और दूसरा उस तरफ है. ठीक है. So these observations that particular histone modification have high probability of occurring at specific functional regions of chromatin has led to the hypothesis that histone tail modification constitute a biological code that can be written, read and erased by specific proteins in the cell. तो इससे हमें क्या पता चला है these are the modifications. They are so specific to with respect to each histone proteins or the histone octamer, the core proteins, that it is well being crypted. इसका मतलब क्या हो रहा है कि ये एक तरह से कोडिंग की तरह प्रेजेंट है इनसाइड द न्यूक्लियस, ठीक है। तो इसको पढ़ा जा सकता है अगर इस तरह की मॉडिफिकेशन है, सो यू कैन अंडरस्टैंड दैट ये मॉडिफिकेशन हाँ जीन एक्सप्रेशन शुरू करेगा या ट्रांसक्रिप्शन डिप्रेस करेगा या ये न्यूली सिंथेसाइज्ड हिस्टोन मॉलिक्यूल्स है। तो ये सब आपको पता चल सकता है वंस यू नो व्हाट काइंड ऑफ मॉडिफिकेशंस आर टेकिंग प्लेस ऑन द हिस्टोन प्रोटीन्स, ठीक है। So that's all about the today's lecture about the histone tail modification. What are the types and at what location it is happening? So guys, if possible, try to remember all these because it will be a very important part to remember. And they might ask in the part B sections of your CSA exams. So stay tuned with the Future Academy. If you like the video, please share and subscribe with your friend circle. Thank you.